Hello, my name is Eleni Livanou, and I am currently a postdoc at the Hellenic Center for Marine Research in Crete, Greece. The project I am presenting today is entitled Modeling Nanoflagellates Bacteria Interactions in the Oligotrophic Eastern Mediterranean Sea. The Eastern Mediterranean is one of the most oligotrophic ecosystems in the planet. This system is characterized by extremely low inorganic nutrient concentrations in the euphotic zone. The scarcity of inorganic nutrients results in a very low values of chlorophyll A, less than 0.05 milligrams per cubic meter, in the southeast part of the basin here, which is the area of interest of the present study. A specific trait of the area is the uncommonly high nitrogen to phosphorus ratio with reference to the typical Redfield ratio, which indicates that phosphorus is the primarily limiting nutrient for the growth of phytoplankton and heterotrophic bacteria in the area. The microbial food web dominates and determines the fluxes of carbon for most of the year. In this system, cyanobacteria and small, less than 5 microns pigmented nanoflagellates are the dominant primary producers. During the stratification period, around 37% of their production is excreted as dissolved organic carbon, and it is channeled into the so-called microbial loop, where it is consumed by heterotrophic bacteria. Bacteria are in turn consumed by heterotrophic nanoflagellates that will be further consumed by higher trophic levels. Moreover, pigmented nanoflagellates have the ability not only to photosynthesize, but also to consume heterotrophic bacteria. This mode of nutrition is termed mixotrophy. However, the functional role of mixotrophic pigmented nanoflagellates in the Eastern Mediterranean microbial loop is rather unexplored. With the aim to better understand the nanoflagellates bacteria interactions in the Eastern Mediterranean, we performed a series of in situ experiments of nanoflagellates grazing on heterotrophic bacteria in the area south of Crete during the onset of thermal stratification when phosphorus concentration was very low. Our results showed that pigmented nanoflagellates, the red bars, contributed significantly to the total bacteria consumption by nanoflagellates at the deeper layer of the euphotic zone sampled, the 75 meters, whereas at the 5 meters, their contribution to bacteriovory was less pronounced. Interestingly, a significant negative correlation was found between the grazing rates of pigmented nanoflagellates on heterotrophic bacteria and the concentration of inorganic phosphorus. Additional nanoflagellates grazing experiments were performed during the late stratified season with surface water from the Cretan Sea when phosphorus concentration is very low. In the beginning of the experiment, we added inorganic phosphorus in 3 microcosms units, while we kept the other 3 microcosms as the control, where no manipulation was performed. The experiment ran for 5 days, and on 3 occasions we performed grazing experiments. Our results showed that in the microcosms where phosphorus was added, a significant reduction of the grazing rate of pigmented nanoflagellates was observed, whereas heterotrophic nanoflagellates remained unaffected by the phosphorus addition. Heterotrophic nanoflagellates in blue, pigmented nanoflagellates in red. These results confirm that phosphorus deficiency induces mixotrophy among pigmented nanoflagellates in the Eastern Mediterranean. In the present study, our aim was to qualitatively explore the pathways of phosphorus and carbon flow through the bacteria nanoflagellates link of the Eastern Mediterranean Sea microbial food web under varying availability of phosphorus. For this reason, we built a model that simulates a simplified microbial food web, and we led the model to reach steady state in order to simulate the stratified phosphorus-depleted upper mixed layer that typically occurs during summer, early autumn in the Eastern Mediterranean Sea. Then we forced a 50-fold increase of the steady-state concentration of inorganic phosphorus, and we let the model run for 5 days after this phosphorus addition. The zero-dimensional mathematical model that was implemented simulates the dynamics of pigmented and heterotrophic nanoflagellates and heterotrophic bacteria. The model takes into account bacteria consumption among pigmented nanoflagellates through mixotrophy and the physiological process of the dissolved organic matter released by pigmented nanoflagellates. Moreover, based on previous studies, heterotrophic bacteria are assumed to be more efficient in phosphate acquisition than pigmented nanoflagellates. The environment is modeled with 8 state variables. the inorganic carbon and phosphorus, the dissolved organic matter, which is expressed in carbon and phosphorus units and partitioned into two compartments. The labile dissolved organic matter compartment is rapidly taken up by heterotrophic bacteria, whereas the semi-labile dissolved organic matter compartment is more resistant to rapid microbial degradation. Finally, dead organisms are added both to the labile dissolved organic matter pool and to the particulate organic matter pool, which is expressed in carbon and phosphorus units. Our model for mixotrophic pigmented nanoflagellates is based on the dynamic energy budget model for photosynthetic cells. The gray frame corresponds to the interior of the cell. The total biomass of the organism is described by four state variables: the structural biomass V, the generalized reserves E, and the distinct reserves of carbon and phosphorus. All transformations of substrates are performed with the concept of the synthesizing unit that is depicted with circles in the diagram. First, the organism takes up inorganic carbon and produces organic carbon via the light and dark reactions of photosynthesis. The organic carbon is combined with phosphorus in a parallel and complementary transformation to produce the generalized reserves E. The substrates that cannot be used directly for the synthesis of generalized reserves due to stoichiometric constraints are stored to the distinct reserves of carbon and phosphorus. Finally, all reserves will be used for the synthesis of structural biomass after maintenance costs of structural biomass have been paid. Pigmented nanoflagellates are the main producers of dissolved organic matter through two physiological mechanisms. First, a fraction of the reserves that cannot be used for the increase of structural mass due to stoichiometric constraints is excreted to the environment as dissolved organic carbon and/or phosphorus. Second, during growth and the transformation of reserves to structure, there are some growth overheads that are released to the environment as dissolved organic matter. The produced dissolved organic matter associated with growth overheads contains both carbon and phosphorus in a fixed stoichiometry and is assumed to be labile. The produced dissolved organic matter associated with the rejected catabolic fluxes from the reserves is assumed to be semi-labile and it has a variable stoichiometry. Evidently, when the organism is growing under phosphorus limitation, this semi-labile dissolved organic matter will be rich in carbon, which is the non-limiting substrate. Finally, dissolved organic matter may be released due to cell lysis. On the basis of the experimental evidence for pigmented nanoflagellates in the Eastern Mediterranean, presented earlier, pigmented nanoflagellates are modeled as type 2A mixotrophs according to the classification system proposed by Stoker in 1998. In this type of mixotrophy, phototrophy, the green pathway, presented earlier, is the dominant and preferred mode of nutrition, whereas phagotrophy and thus bacterial consumption, the red pathway here, is initiated in response to inorganic phosphorus limitation. Inorganic nutrients and nutrients obtained from prey are processed in parallel by the synthesizing unit highlighted in blue, and they are considered substitutable, meaning that either of them combined with carbohydrates can form the generalized reserves. In type 2A mixotrophs, photosynthesis is obligatory. Therefore, only if the synthesizing unit has already bound carbohydrates, then it can also bind nutrients obtained through phagotrophy to produce generalized reserves. Finally, because type 2A mixotrophs prefer phototrophy over phagotrophy, the synthesizing unit will reject nutrients from prey in case inorganic nutrients arrive at the synthesizing unit. In order to investigate how the carbon and phosphorus fluxes are altered due to a pulse of phosphate added to a phosphorus-limited system, we use alluvial diagrams. In these diagrams, the sources are placed to the left, here, and the sinks are placed to the right, while the width of a line connecting the state variables is proportional to the magnitude of the flux between the two compartments. Bacterial carbon is equally consumed by heterotrophic and pigmented nanoflagellates before phosphorus addition, as shown by the red arrows, whereas after phosphorus addition, heterotrophic nanoflagellates are the major consumers of heterotrophic bacterial carbon. Moreover, pigmented nanoflagellates are the major producers of semi-labile dissolved organic matter before and after phosphorus addition, as shown by the green arrows. However, the allocation of carbon to the semi-labile dissolved organic carbon compartment by pigmented nanoflagellates is diminished after phosphorus addition, as shown comparing the width of the green line here and here, before and after phosphorus addition. Regarding the phosphorus fluxes, as shown by the blue arrows, pigmented nanoflagellates have a minor contribution to the consumption of inorganic phosphorus before phosphorus addition. But after phosphorus addition, the role of pigmented nanoflagellates in the inorganic phosphorus consumption is notably enhanced, as shown by the width of the blue line here. This coincides with a considerable decrease in the contribution of bacterial phosphorus to the total phosphorus uptake flux by pigmented nanoflagellates after phosphorus addition, as shown by the red arrows. And it is evident when comparing the width of the red line here after phosphorus addition and here before phosphorus addition. Sankey diagrams were used to show how carbon and phosphorus taken up from the environment are allocated to the different metabolic pathways within pigmented nanoflagellates before and after phosphorus addition. In these diagrams, inputs are placed to the left. Metabolic losses due to excretion processes are depicted as arrows pointing up, and the remaining flux available for growth after the metabolic costs have been covered is depicted with an arrow pointing to the right. Regarding carbon flux, before and after phosphorus addition, most of the carbon taken up by phytoplankton is through photosynthetic carbon fixation, while bacterial carbon does not contribute significantly to the total carbon uptake. Nevertheless, phosphorus addition causes a decrease of the relative contribution of bacterial carbon to the total carbon uptake flux in pigmented nanoflagellates. Before phosphorus addition, most of the carbon taken up is exuded as semi-labile dissolved organic carbon. On the contrary, after phosphorus addition, the percentage of carbon that is lost as semi-labile dissolved organic carbon decreases sharply, and the percentage of assimilated carbon that is allocated to growth increases almost twofold, here before phosphorus addition and here after phosphorus addition, since when there is no phosphorus limitation, the photosynthetically produced organic carbon can be used for cell growth. Looking at the phosphorus budget of pigmented nanoflagellates during the phosphorus depleted period, we see that most of the phosphorus needs of pigmented nanoflagellates are covered by heterotrophic bacteria consumption. Instead, as soon as phosphorus is added to the system, most of the phosphorus needs of pigmented nanoflagellates are covered by inorganic phosphorus uptake. To sum up, our model simulations pinpoint the existence of an alternative microbial loop in the Eastern Mediterranean, which operates in a phosphorus-dependent way. According to our model simulations, during the phosphorus-depleted period, most of the carbon fixed by pigmented nanoflagellates is excreted as semi-labile dissolved organic carbon via the active exudation mechanism. Thus, pigmented nanoflagellates promote phosphorus uptake by heterotrophic bacteria and support bacterial production. At the same time, pigmented nanoflagellates consume bacteria via mixotrophy to obtain the scarce phosphorus in a particulate form. Thus, mixotrophy allows pigmented nanoflagellates to circumvent the P limitation and enables them to survive in the ultra-oligotrophic environment of the Eastern Mediterranean. However, when the inorganic phosphorus concentration increases, the significance of this alternative microbial loop is reduced, since pigmented nanoflagellates lower their grazing rate on heterotrophic bacteria and consume the available inorganic phosphorus. Therefore, under phosphorus replete conditions, the relative contribution of the heterotrophic nanoflagellates to bacterial grazing and the classical microbial loop are enhanced. Thank you, and this is the end of the presentation.